Very good morning to all. As a part of paper 1, we are discussing the topics of virus, bacteria, and fungi. These 3 topics we are discussing now. Yes or not? Of that, till now we have studied the 3 units of the fungi. Fungi, thallus organization, general characters, and what you have studied? Fungi, general character, thallus organization, nutrition, reproduction, classification, economic importance. These all we have studied. Yes or not? Now, today we are going to discuss the second unit, that is bacteria. Bacteria, general character, starter, general characters, and size, shape, reproduction, classification, and economic importance of bacteria. That we are going to discuss. Okay. You know the bacteria. What is it? The bacteria. Have you ever seen the bacteria? Have you ever heard or not? You have studied in intermediate degree also. Bacteria. Bacteria, you know, who first discovered, identified the bacteria? Who first discovered, identified the bacteria? Anybody know? You know, Anton van Leeuwenhoek first observed the microbes on his sputum. అతను ఉమ్మి లోపల sputum ఉమ్మి లోపల first he has taken out a sputum and observed under the microscope. He observed the animals, that means small minute organism which are moving. That's why given the name it has, what the name he has given? Do you know? Animalcules. Animalcules. Given the name as animalcules. Has given the name for that as an animalcule. You know the what does it mean? The bacteria meaning in the Greek or Latin? Bacteria means a small rod in the Greek. You know what does it mean? The study of bacteria. Study of bacteria is called as study of bacteria. Bacteriology. Study of bacteria is called as bacteriology. Oh, he first developed the microscope and studied the microorganism. Describe the bacteria East protozoa, which we can see under the microscope only. What he has given the name it as? Animalcules. What does it mean? What the name he has given? Animalcules. Given the name it as animalcules. They are small, minute, and very small and minute that are showing the movement as like animals. That's why given the name it as animalcules. So many exams this question will ask. Animalcules. Leeuwenhoek, Anthony Leeuwenhoek, given the name it as animalcules. And who given the term bacteriology? Do you know who given the term bacteriology? Bacteria name given by whom? Anybody? Anybody know who has given the name as bacteria? Who has given the name it as bacteria? Anybody know? You know Herrenberg. Herrenberg given the name as bacteria to the animalcules which are first identified by whom? Leeuwenhoek. Leeuwenhoek first identified the bacteria, yeast, protozoans, and he first observed the bacteria under in sputum and named it as animalcules. That animalcules name changed it as a bacteria, given the name bacteria by Herrenberg. H-R-E-N-E-B-R-G. Herrenberg. Okay, Herrenberg given the name, the term bacteria in 1938. You know the Louis Pasteur? Louis Pasteur? You know the Louis Pasteur? Pasteurization technique. Still now we are using that pasteurization technique. 19th century pasteurization techniques, 20th century pasteurization technique. Still now we are using. What the pasteurization technique, you know? Anybody is there? Pasteurization technique. The French scientist Louis Pasteur. Louis Pasteur. You know the Louis Pasteur? Louis Pasteur. Louis Pasteur given the name it as what he has done. The Louis Pasteur described the new identified the new technique that is do you know pasteurization technique. You know milk, for the milk preservation we heat the milk up to 78 degrees centigrade and then cool down into 10 degrees centigrade, preserve in the cold temperature so that milk will be preserved for the long time. Otherwise, the very next day, milk will be spoiled by the microbes. To maintain the purity of the milk, the Louis Pasteur identified the new technique that is pasteurization. Pasteurization technique. That pasteurization technique is useful for the preservation of the milk. And you know, Louis Pasteur, one more thing. He also discovered, invented the vaccine for the rabies, dog bite. For the dog bite disease, rabies vaccine also invented by Louis Pasteur. No, that's why we call the Louis Pasteur as a father of modern bacteriology. You know, Anthony van Leeuwenhoek is called as father of microbiology. Father of microbiology. Louis Pasteur and also Hook is called as father of modern microbiology or modern bacteriology. Modern microbiology are also called as father of bacteriology. Father of bacteriology. You know, the bacteria are prokaryotes or eukaryotes? All the, yes, all the bacteria are prokaryotes. Prokaryotic. Prokaryotic means, what does it mean, the prokaryotic?
Lack of nuclear membrane.
They? Pro means pre, fast. Karyotic, before the nuclear organism. That means there is no specific nuclear membrane, nucleus, nucleolus. Only nucleides, only genetic material is present. As DNA, RNA, protein, the genetic material only present. Membrane is absent. Nuclear membrane is absent. Nucleolus, nuclear membrane, histone protein, absent. There is no protein binding DNA. Only naked DNA is present. That we calling it as prokaryotes. You know, bacteria, archaebacteria, the first bacteria are archaebacteria. This can survive in hard core situations, conditions, very hot condition and very cold conditions, also very alkaline condition. Even Methanococcus bacteria, methane gas releasing animal excretory material. The hardcore bacteria, archaebacteria, eubacteria, and you know the cyanobacteria, blue-green algae. These three type of the bacteria are there: archaebacteria, eubacteria, and cyanobacteria. Okay, these three are there. Archaebacteria are very primitive bacteria. It having the very thick coated NAG-NAM coats are there. N-acetyl glutamic acid, N-acetyl muramic acid, murate. Thick coated cell wall is present to overcome the abnormal conditions in the environment, very hard condition in the environment. Okay. And you know who is the father of microbiology? Who is the father of bacteriology? Who given the name term bacteria? These all are very, very important. You should know the basics. These all the basics you should know. Otherwise, there is no use of studying the bacteria. Huh? Leeuwenhoek, the father of microbiology, and Louis Pasteur is the father of modern microbiology or bacteriology, modern bacteriology. Who given the name bacteria? Who given the name bacteria? Ehrenberg. Ehrenberg. E-H-R-E-N-E-N-B-E-R-G. Ehrenberg is the given the name of bacteria to the animal. Have given the name to the bacteria as animalcule. Ehrenberg given the name it as bacteria. Ehrenberg given the name it as bacteria. You see, bacteria is eubacteria and remaining are eukaryotic. All the eukaryotics, algae, fungi, plant, animalia, these all are comes under eukarya. These two comes under prokaryote. These two comes under prokaryotes. Okay, and you can see the variation between the among the plant cell, animal cell, and the bacterial cell. You see, what are the variation? You see, the plant cell, animal cell, and bacterial cell. You know, the animal cell, the cell wall is present or absent? Are you there? In the animal cell, cell wall is absent. Whereas in the plant cell, cell wall is present. You see the cell wall. Cell wall is present. Cell wall is made up of it, cellulose, pectin. And you see in the bacteria, cell wall is present. Bacterial cell wall is made up of it. Anybody say? Bacterial cell wall is made up of it.
Peptidoglycans.
Yes, peptidoglycans. Also called as mucopeptides. Peptidoglycans are mucopeptides. You see the variation between the plant cell, animal cell, and bacterial cell. Plant cell, animal cell, there is no cell wall. Cell wall is absent. In the plant cell wall is present. Even in the bacteria also, cell wall is present. Cell wall is made up of it, peptidoglycans. You know, why we are not included the bacteria in the animal? Why we included in the plants? Just now I say. Cell wall. Presence of cell wall. Reproduction by binary fission. Presence of cell wall, reproduction by binary fission, endospores, cysts, as like the plants. That's why the bacteria are included in the in which
Plants.
plants, not in animals. Okay, you should know why the bacteria are included in the plants, why not in the animals? Why? Just now I said now, cell wall and the reproduction as like the plants, having the cell wall and reproduction as like a plants. That's why the bacteria are included in the plant, not in animals. Not in animals, included in the plants. Okay. Whereas, you know, the bacteria, ultra section of the bacteria cell, we can discuss. You see in the plant cell, all the membrane bounded cell organelles are present. Chloroplast, mitochondria, endoplasmic reticulum, Golgi bodies, nucleus, ribosome, lysosome, spirosome, galaxosome, peroxisomes, all are present. Here in the animal cell also all are present except chloroplast. Except chloroplast, remaining all are present. Here in the bacteria also, what are there? Mitochondria, chloroplasts are absent, endoplasmic reticulum, these all are absent. So membrane bounded cell organelles are absent in the bacteria. That also very important. Membrane bounded organelles. That means chloroplast, mitochondria, Golgi bodies, endoplasmic reticulum. These all are present or absent? Present or absent? Nobody is responding. Absent. Ribosomes are present. Only nucleide, naked DNA, nucleide is present. Plasmids are present. Okay. But membrane bounded cell organelles are absent in the bacteria. That you should know difference between the plant cell, animal cell, and bacterial cell. Why included in the plant? Why not in the animal? That all you should know before studying the bacteria. Okay. Okay. Are you there? Who is the father of bacteria? Bacteriology. Study about the bacteria is called as bacteriology.
Bacteriology.
Who given the name bacteria?
Ehrenberg.
Ehrenberg. Yes, good. Okay. And next, you know the Dutch scientist Robert Hooke. Robert Hooke, you know, the first identified the cell. Living, you know the difference between living cell and non-living cell? Huh? Okay, at least you know the difference between prokaryote and eukaryote?
So nuclear membrane is absent in prokaryotes, it is present in eukaryotes.
Yes.
And the membrane bound organelles are present in absent in prokaryotes.
Prokaryotes present in eukaryotes. Good.
Eukaryotes.
Just now I told. Again, our or more.
Types of ribosomes also.
Yeah, ribosomes are present. Ribosomes are present in prokaryote and eukaryotes, but cell membrane organelles are cell membrane organelles are absent. And you know there is no chromosome structure in the prokaryote. Okay, only single coil chromatids are present. Where the annotation? Single coil chromatids are present. That means nuclei. Only single coil chromatids are present. There is no chromosome structure. Chromosome structure, specific DNA chromosome structure is absent in the prokaryote, present in the eukaryote. Histone proteins are absent in the prokaryote. Histone proteins are present in the eukaryote. What is the use of histone proteins? Histone proteins which bind the DNA, it forms as like a bead chain, Pusara danda, bead chain, Pusara danda antama, as like a bead chain. Histone protein which bind the DNA. There is a 8 histone protein that also asks in so many questions. How many types of histone proteins are there? 8. H1, H2A, H2B, H3, and H4A, H4B, H5, H6. These all are 8 type, interconnected and intraconnected histone proteins, so that it forms the chromatid structure in the eukaryotes, but it is not there in the prokaryotes. That's why it's not present. You know, the cell wall of the prokaryote is made up of it peptidoglycans. And the cell wall of, you know, the plant cell wall is made up of it cellulose. Fungal cell wall is made up of it chitin. But there is no cell wall in the animals. No cell wall in the animal. Just now we have seen the pictures. Differences between the plant cell, animal cell, and bacterial cell. Okay. No cell wall in the animal cell. The plant cell wall is made up with cellulose. Fungal cell wall is made up with chitin. Okay. No cell wall in the animal. Bacterial cell wall is made up with peptidoglycans. Okay. And then what is the other difference? At least you read out, you will remember for the long time. You see, the mitotic divisions are absent in the prokaryote and present in the eukaryote. Okay. I know 70s ribosomes only present in the prokaryotes and 80s type of the ribosomes are present in the eukaryotes. And especially ribosomes are attached to the endoplasmic reticulum. Endoplasmic reticulum attached with the ribosomes. Endoplasmic reticulum attached with the ribosomes in the prokaryote. That's why we call it a rough endoplasmic reticulum. This will be useful in the protein synthesis. In the protein synthesis, rough endoplasmic. Why rough? Ribosomes are attached to the endoplasmic reticulum. That's why it is a rough, not smooth. Smooth means if there is no ribosome, that is smooth. If the ribosomes are attached, that is rough. You know 70s and 80s ribosome? Each ribosome having the two subunits, the larger subunit and smaller subunit. Do you know or not? 50s, 30s subunit. Two subunits combinedly form the 70s ribosome. These are there in the prokaryote. Whereas in the eukaryote, 60s, 40s. 60s big subunit and 40s small subunits combined, it forms the 80s type ribosomes. 80, yes mean Swedberg. Yes, rotations. How many rotations per second? 80 years and 70 years. Okay, based upon their rotation in the centrifugation. Like 70 years and 80 years. 70 years are present in the prokaryote, 80 years are present in eukaryote. Okay, and then flagella. Flagellas are usually present in negative bacteria. If even present, it is made up with flagellin protein. Made up with flagellin protein. Whereas in the other algae, fungi, bryophyte, pteridophyte, gymnosperm, angiosperm, the flagella is made up with microtubules, that is 9 plus 2. 2 flagellas at the middle, 2 pimples, and it surrounds with the 9 pimples. 9 plus 2 arrangement is there in the eukaryotes. We have studied in the algae, even in the fungi, Phycomycetes fungi, Mastigomycotina. You know, the membrane bounded cell organelles, mitochondria, Golgi bodies, endoplasmic reticulum are absent in prokaryote. In prokaryotes, these all are present in eukaryotes. You see, lysosomes also absent in the bacteria but present in the eukaryotes. You know what we call the lysosome? What is the use of lysosome? Lysosomes, we call it as suicidal bodies of the cell. Suicidal bodies of the cell. Which one? Which one we call it as the suicidal bodies of the cell?
Lysosomes.
Lysosomes, which digest the poisonous cell organelles if any there, if any degraded, if any diluted, that all will be digested by the lysosome to prevent from the damage, of the whole cell. Phagocysis will take place. That's why we call it as suicidal bodies. Ribosomes are the protein synthesized bodies. Lysosomes are the suicidal bodies. Ribosomes are useful for the protein synthesis. Which organelle is causing for the protein synthesis in the cell? Ribosomes. Okay. And then the Golgi operators, it form the vesicles, cisternates are present. Golgi bodies, that Golgi bodies are useful in the formation of ribosome, lysosomes, pyrosome, galaxosomes, all the micro cell organelles formed from the Golgi bodies secretion. Okay, that are absent in the prokaryote, present in your home, present in eukaryotes. Okay, these are the difference between prokaryote and eukaryote. This question asks a short answer question. What is the difference between prokaryote and eukaryote? What is the difference between prokaryote and eukaryote? They ask the question. These differences you have to write. Okay. What are the difference between prokaryote and eukaryote? Can you say? What are the difference? You repeat, read out, so that you can remember. What are those? Nucleus absent and present in the eukaryotes. Okay. Membrane bound cell organelles are absent and membrane bound cell organelles are present. Chromosome chromatid are absent and chromatids, multiple number of the chromosomes are present. Histone proteins are absent, histone proteins are present. And the cell wall is made up with peptidoglycan and cell wall is made up with cellulose or chitin. Chitin in fungi, cellulose in all the plant cells, absent in animal cells. And mitotic division absent in the prokaryote, present in the eukaryote. Ribosome, 70S ribosomes are present in prokaryote, the 80S ribosomes are present in eukaryote. Flagellas are present in negative gram-negative bacteria and it made up with flagellin protein. Whereas in the plant cells, flagella is made up with microtubules, that is 9+2. And cell membrane bonded cell organelles, mitochondria, lysosome, Golgi bodies, endoplasmic reticulum are absent in the prokaryote. These all are present in the eukaryote. These are the variation. These are the difference. These are the distinguishing character between the prokaryote and eukaryote. Okay. U means true. Karyote. True nucleus is present. Primitive nucleus is present. Okay. Size of the bacteria. Can you say which one is the smallest bacteria? Which one is the largest bacteria? Can anybody say? Which one is the smallest bacteria? Can anybody? Do you know? Can anybody? The Pachyderella. Pachyderella is the smallest bacteria. The counted exam in the general knowledge point of view also you should know which one is the smallest bacteria. Which one is the largest bacteria? Which one is the largest bacteria? Bigieta. Bigiata is the largest bacteria. This is sulfur bacteria. It Bigiata is the Bigiata is the read out. Tell me what it is. Bigiata is the that we use in the cell biology studies. This all you should know. Okay. And then you can see the shape of the bacteria. What are the shapes of the bacteria? What are the shapes of bacteria? Read out, Amma. You respond. Otherwise, it will be mono action. You should know, na? You see how many types, how many shapes of the bacteria? What are the shapes of bacteria? Bacteria shapes. This also short answer.
Focus, Bacillus, Vibrio,
Ah, ah,
Spirillum.
spirillum. Okay.
Short
If it is
shape.
a raw, yeah, if it is a round circular, that we calling it as
Coccus.
coccus. Round circular, that we called it as coccus. Okay, cocci. Single one is the cocci. Okay, cocci. If it is a two cocci bacteria, that we call a diplococcus.
Diplococcus.
You know the diplococcus pneumoniae that causing pneumonia disease
Yes.
to our human beings. That is diplococcus pneumoniae. If it is a chain-like structure, that is streptococcus.
Streptococcus.
If the more number of the cocci are attached, that is streptococcus. If the more number of the cocci arranged in uneven shape in the cluster, that we call it as staphylococci. Staphylococci. Cocci, diplococci, streptococcus, staphylococcus. Even single coccus again, 4 types. What is there? Monococcus, single. Monococcus. If two, diplococcus. If the more in chain, streptococcus. If the more in uneven shape, cluster shape, that is staphylococcus. Okay. That are the types, shapes, structures. And then the next is bacillus. That means rod shape. Bacillus, rod shape. Okay. You know, our lactobacillus. This lactobacillus is rod shape. You know the lactobacillus. It ferment the milk
Curd.
and it convert into curd. For the preparation of curd, we add the lactobacillus. You know, we all are the biotechnologists. We are doing the biotechnology in our daily life. We are adding the curd to the milk so that all the milk will be turned into curd. That is the biotechnology product. We are using the bacteria, lactobacillus, for the preparation of curd. Okay, like same cocci, it's also bacilli, diplobacillus, streptobacillus, and staphylococcus. Okay, bacilli, diplobacillus, streptobacillus, and staphylococcus. Like cocci, four types. Bacillus also four types are there. Okay, and then you can see the vibrio. Vibrio means comma shape. You know the comma full stop, comma, comma full stop. It's look like a comma, curved. Curved bacteria are called as vibrio. You know the Ganga gel. Why we call it a fresh, pure water? Do you know why we feel it as a fresh water? Ganga gel, pure water, Shuddha gel. Can anybody? Can anybody? Can anybody say? Respond. If you don't know, you say don't know. That's it. You don't waste the time.
Don't know, sir.
Okay. You know the in the Ganga gel, Ganga water, there is a Dilobvibrio bacteriovirus. Dilobvibrio bacteriovirus. Bacteria is there. It is a bacteria. It is hyperparasite upon bacteria. It is a bacteria. It can parasitize upon bacteria, so that it kill all the microbes, bacteria, microbes in the Ganga water. That's why Ganga water will be purified by this Dilobvibrio bacteria. That's why if Ganga gel is pure water, even with contaminated with all the microbes or debris, it will be purified in the Ganga water. That's because of this bacteria. Which one? Vibrio, comma shape bacteria. Comma shape.
Vibrio bacteriovirus.
Dilobvibrio bacteriovirus. Bacteriovirus. That means it eats the bacteria. Bacteriovirus. Eat the bacteria. Dilobvibrio. Vibrio, comma shape. Two comma shaped bacteria. Dilobvibrio bacteriovirus, which is present in the Ganga water. That's why is purified purified from the all the microbes. You should remember the Ganga gel bacteria. Which bacteria is present in the Ganga gel? Why we Ganga gel purified water? Huh? Okay, and then the next is spirillum, spiral cell, spirally called spirillum, spirally called spirillum. Okay, and the square shape, square, not the rod shape, square shape. That means square means the four angles are equal. That is square shape. Okay, and star shape. These are the various shapes of the bacteria. These are the various shapes of the bacteria. This question also short answer asked. Write about the various shapes of the bacteria. Bacteria having the different shape that are mainly four types. What are those? Cocci, bacilli, vibrio, spirillum. And all The coccus, if it is arranged in octangular, that we call as sarcina. Another shape, sarcina, sarcina shape. Okay. And again, coccus four types, bacillus four types are there. Vibrio, comma shape, spirillum, spiral, square, star shape. Various shapes of the bacteria are there that we have discussed. Okay. And you can see the various shapes of the bacteria shape. You can see here the cocci, diplococci, and you see tetrad and sarcina shape structure. You see micrococci, streptococci, staphylococci, uh, diplococci, and tetrads. If the four cocci are present, and you see the rod shape for bacillus, diplobacillus, streptobacillus, and staphylobacillus. You see the various shapes of it, strepto and staphylo, spore forming rods and branched, and you can see the spirillum, vibrio shape. Okay, these all are the various shapes of the bacteria. Okay, and then we will discuss the bacterial cell structure. Bacterial cell structure, ultra structure of the bacterial cell. Yes, a question. Ask the question, ultra structure of the bacterial cell. Ultra structure of the bacterial cell, very, very important. What are there in the ultra structure of the bacteria, that all discussed in this ultra structure of structure. The first uppermost outer layer, gelatinous layer, is called as capsule. Capsule. Capsule is the outermost layer. This capsule is present in the gram-positive bacteria. Capsule is present in gram-positive bacteria. Okay. This is the mucilaginous layer. It's made up of with mucilage. The outer, that means above the cell wall, mucilage, extra layer is present. This extra layer is calling it as capsule. It looks like a capsule covering sheath. It looks like a covering sheath. That's why we call it as capsule. It is made up of it polypeptides, polysaccharides. Made up of it polypeptides and polysaccharides. This is present in that cause for the fermentation, alkalization of our food to easy digestion, for the easy digestion. That E. coli is present in our intestine, that is useful for the easy digestion. Okay. Gram staining technique. Do you know the gram staining technique? Gram staining. You have done the gram staining in the practical. Whether you are done or not, you have done the gram staining technique. That gram staining technique to differentiate, to distinguish the bacteria, to distinguish the bacteria, gram positive and gram negative bacteria. To distinguish the gram positive and gram negative bacteria, we use this gram staining technique. We use that gram staining technique. That gram staining technique will be useful to distinguish gram positive and gram negative bacteria based upon their cell wall component. Based upon their cell wall component, we can easily classify the bacteria into gram positive and gram negative bacteria by using the gram staining technique. That gram staining technique first done by Christian Graf. Christian Graf. That's why given the name as gram staining technique. You know the crystal violet color they use for the staining. Crystal violet color. Crystal violet color used for the gram staining technique. The crystal violet color, iodine, alcohol, acetone, these all are chemicals which we use in the gram staining technique. Which we use in the gram staining techniques. Okay, that are very, very useful to identify and distinguish gram positive and gram negative bacteria. Gram positive and gram negative bacteria. Gram positive and gram negative bacteria. It is very easy. Oh, screen share is off. Huh? Gram positive and gram negative bacteria identification. It is very easy by gram staining technique. It is a simple staining technique. It is a simple staining technique to stain the bacteria, to distinguish the bacteria, gram positive and gram negative. You can see the cocci bacteria, bacillus bacteria. Could you able to see this?
Yes, sir.
Okay. You see the cocci bacteria, actual colorful pictures. Diplococcus, Streptococcus, Staphylococcus, Sarcina, tetrads. The 4 are presented tetrad. If the 8 or more in the round ball shape, that is Sarcina, Sarcinate, and Streptococcus, Staphylococcus, uneven arrangement. You see the bacillus, Diplobacillus, Streptobacillus, and Staphlobacillus. And you see the palisade type of the bacteria, palisade shape. And you see are you hearing? Hello? Is it audible? Hello? Hello? Is it audible? Are you
Yes, sir.
there?
Yes, sir. Audible. Yes, sir. Audible.
Okay, okay then. Yeah, these are the various shapes of the bacteria we have studied. The very, very important I told you already. And then these are the very special shape of the bacteria also there, along with the normal shape, star-shaped bacteria and filamentous bacteria. You know the filamentous bacteria In which group? Filamentous bacteria. You know the actinomycetes. Actinomycetes bacteria are filamentous shape. That means it look like a fungi, fungal filamentous-like structure. Fungi, filamentous-like structure. You see here the first picture. Fungal filamentous-like structure. It look like a fungi, but it is a bacteria. That we call a actinomycetes. Most of the actinomycetes fungi. That means actinomycetes, that bacteria. Actinomycetes are bacteria. These are gram-negative bacteria, which are look like a fungi that we are using for the antibiotics productions. Antibiotics, streptomycin, conomycin, tetramycin, oxytetracycline, even lactobacillus. These all are comes under streptoactinomyces bacteria, filamentous bacteria. Okay, you can see the various shape. Pleomorphic bacteria, that means they change their shape, size, structure, not in the unique constant structure. You see the mycoplasma. Mycoplasma is also one of the bacteria having the highest quantity of GC content, guanine cytosine content. Mycoplasma. You know the mycoplasma? Cell wall-less. Competitive exam lo so many times they ask the question. Cell wall-less microorganism. No cell wall. Cell membrane, cell wall is absent only. Cell membrane, plasma membrane is present. Very minute, cell wall-less microorganism is mycoplasma. That is very important. In any exams, any competitive exam is very, very important. Okay. You see the various shapes, star shape, vacuole shape, uneven shape, pleomorphic. And then the next is outermost layer of the bacteria cell is calling it as outermost layer of the bacteria, outermost layer of the bacteria cell. It is made up with gelatinous. It is made up with gelatinous structure that made up with polysaccharides or polypeptides. Polysaccharides and polypeptides. It's slimy, gelatinous. It forms the capsule-like structure. That's why we call it as capsule. Outermost layer of the bacteria, just above the cell wall. Just above the cell wall, outermost layer of the bacteria cell is what it is
Capsule.
capsule. Okay, that capsule, fortunately that capsule is present only in the gram positive bacteria, not in the gram negative bacteria. You know the gram positive bacteria are useful bacteria, gram negative bacteria are harmful bacteria. Okay, most of the gram negative bacteria cause the diseases. Most of the gram positive bacteria synthesize antibiotics, useful in our daily life, in the food, agriculture, industry, and so many, so forth. Okay. So that it can be prevented from the adverse conditions by forming the capsules. You know the cell wall. The cell wall is the outermost layer of the cell, just below the capsule. That is made up of it. Cell wall is made up of it. I told you the cell wall is made up of it.
Chitin.
Chitin. Chitin in fungi. Fungal cell wall is made up of it. Chitin. Here peptidoglycans. I told you fungal cell wall is made up of it. Peptidoglycans. Peptides and glycans. Peptidoglycans are present. Along with that, in the gram-negative bacteria, more number of the phospholipids, lipopolysaccharides are present. More number of the thick-coated lipopolysaccharides are present in the gram-negative, along with the peptidoglycans. Along with the peptidoglycans, lipopolysaccharides also present in the gram-negative bacteria, not in the gram-positive bacteria. Gram-negative bacteria having the thick-coated lipopolysaccharides along with the peptidoglycans. Along with the peptidoglycans. That you should remember. That's why we can easily distinguish the gram-positive and gram-negative bacteria by staining. You know, especially the cell wall is made, I told you, cell wall is made up of it NAG and NAM. NAG and NAM. N-acetyl glutamic acid, N-acetyl muramic acid. N-acetyl glutamic acid and N-acetyl muramic acid. These are due to this, it having the hard core cell wall. It will be prevented. These type of the peptidoglycans or mucopeptides are very hard, tough coated, so that the bacteria will be prevented. That's why because of NAG and NAM, the bacteria cell wall is very rigid. It shows the very rigidity. Okay. And along with that, I already told you in the gram-negative, lipopolysaccharides are present in the gram-negative bacteria. You know, I already told you the gram staining technique first identified by Christian Gram. By whom? Christian Gram, Hans Christian Gram, 1884. First, I differentiated based upon distinguishing characters of bacteria, based upon their cell wall structure. Based upon their cell wall structure, the bacteria are divided into gram positive and gram negative. Gram positive and gram negative. Positive and negative. Divided into two, that are gram positive and gram negative. Okay. You see, peptidoglycans are very much thick. Up to 80% of the peptidoglycans are present in the gram positive cell wall. And whereas in the gram negative cell wall, peptidoglycans are at the middle. It is covered with the phospholipids on the both the sides. It forms the three layer. And you know, the gram positive bacteria take the purple stain. Gram negative bacteria take the which stain?
Red stain, saffron.
Saffron in the second stain. Counter stain, not the first stain. Okay. Why take the counter stain? Why not the first stain?
Because of lipopolysaccharides outer present over the peptidoglycan.
Yes, lipopolysaccharides over the peptidoglycans in the both the sides, up and lower side, both the sides. That means more number of the lipids. That lipids are dissolved in the crystal violet. So that dissolved in the acetone or alcohol. That lipids are dissolved in the acetone or alcohol. Whenever we pour the acetone or alcohol, absolute alcohol upon the bacteria, automatically these lipopolysaccharides will be dissolved. So that whatever the first stain, crystal violet stain, will be removed in the gram negative, and whatever the second stain, we pour in the bacteria, the second stain will be the remain in the gram negative. First stain will be removed by acetone and alcohol because of more number of the lipids will be dissolved in the acetone or alcohol, so that first stain will be removed. Whenever we add the counter stain, that counter stain will be attached to the peptidoglycan, so that the gram negative will be in the second stain, counter stain, saponin stain color. Whereas the gram positive bacteria is in violet in color. It take the first crystal violet, first stain, crystal violet. There is no lipopolysaccharide layer, so there is no dissolve. Whatever the stain we use, that will be stick to the cell wall, so that it will be in the crystal violet, violet in color. Whereas the gram negative in pink color or chaperone in color, so that we can easily distinguish whether it is gram positive or gram negative. What are the steps which are there in the gram staining? We already done in the practical. What are the steps are there? First we take the bacterial stain. Okay,
after that fixing.
We make it smear by heating.
By heating over the slide.
Heating under the Bunsen burner, whatever it is, in the light, and we make a smear.
And then add a crystal violet.
Add crystal violet, yes. And then
rinse with water after 1 minute.
What?
Rinsing with water after 1 minute.
Rinse with the water. That means we have to pour the water from one side. Rinse means not brush. Okay. It stain whatever the smear must be there. Rinse means not clean all the glass, all the slide. And then add the iodine solution. Iodine solution. And then absolute alcohol or acetone.
Decolorization.
So that whatever if it is a gram negative, whatever the lipopolysaccharide layers are there, that all
it will be washed out.
Dissolved, washed out. And then
adding the saffron.
Adding the second stain, counter stain, chaperone. If it is a gram negative, it take the counter stain. If it is a gram positive, it taken already first stain. First stain, crystal violet stain only will be remain. It is not dissolved. It is there in the gram positive. That's why we can easily distinguish whether it is gram positive or gram negative. Gram positive or gram negative, we can easily distinguish. That is, we have to observe under the microscope this slide, and then we can easily distinguish whether it is gram positive or gram negative. We observe the lactobacillus, so that usually we can identify it as a gram positive bacteria, useful bacteria. Okay, or streptococcus or staphylococcus, streptobacillus, that is equal, that all we can easily identify it as a gram negative bacteria, so that we can easily distinguish. And what are the difference between gram positive and gram negative? I already discussed now. What are there? The cell wall, differentiation, single layer peptidoglycans are present. Here the triple layer peptidoglycans, it surrounds with the lipopolysaccharides at the both side. Okay. Outer membrane, phospholipid membrane is absent in the gram positive, present in the gram negative. Okay. Periplasmic space, present in some of the gram positive bacteria. It's present in all the cells in between the peptidoglycans and phospholipid. There is a periplasmodial space is present. The chemical composition I already told you. Peptidoglycans are more, 80%. More percentage of the peptidoglycans are there. Very low percent of the lipids are present. Here it is opposite. Peptidoglycans are only up to 20%, and lipids are more percentage. high number of the lipids, phospholipids, lipopolysaccharides are present. Okay, you see, these are insoluble in potassium hydroxide solution. These are soluble in gram-negative, soluble in potassium hydroxide solution. Why? It having the lipids, lipopolysaccharide. That's why it dissolve. Okay, rigidity. High rigidity is present in the gram-positive, and elasticity is present in the gram-negative. Why? More number of the lipopolysaccharides. Okay, gram-positive bacteria show the high susceptibility, but gram-negative bacteria show high resistance. Nutritional requirement. What is the difference? Very simple in the gram-negative, hard in the gram-positive. And you know the permeability, more permeability is there in the gram-positive, less permeability is there in the gram-negative. And you know flagellas are present in the gram-negative and absent in the gram-positive. Okay, these all are the variation in between the gram-positive and gram-negative bacteria. In the ultra structure of the bacteria, we are discussing the second one. We are discussing first one capsule, second one cell wall. Based upon the cell wall, we categorize the bacteria into two groups that are gram positive and gram negative. And the next is after the cell wall, the next, you can see the difference between the gram positive and gram negative bacteria. You see, triple layer is present in the gram negative, only single layer is present in the gram positive. Okay. Capsule is present in the gram positive, absent in the gram negative. Okay. And flagella is absent in the gram positive. Flagellas are present in the gram negative. Okay. These are so many variations are there, I already told you. Okay. And then you can see the clear structure of the cell wall between the gram positive and gram negative. You see, there is a three layer phospholipid layers are there, plasma membrane phospholipid layers are there in the both the side. Lipopolysaccharides are more in the gram negative, absent very less in the gram positive. You see, peptidoglycans are more in the gram positive and peptidoglycans are more in the gram positive, very less in the gram negative. Lipopolysaccharides are more in the gram positive, no or less in the gram positive. Okay. And then you can see the protoplast. The plasma membrane, you know the plasma membrane is made up with lipoproteins that is below the cell wall. That is below the cell wall, capsule, cell wall, plasma membrane. You know the cytoplasmic membrane, sometime what happen in some of the bacteria, the plasma membrane will be folded inside, it form a specific structure that we calling it as mesosomes. What we calling it as mesosomes. That mesosomes are present. The plasma membrane it is folded inside and it form the mesosome. It will be acting like as in mitochondria, as like a respiratory organ. Also useful for the binary fission to separate into two bacterial cells, respiratory and reproductive organ, acting like the both, but it is a plasma membrane, infolded plasma membrane that And then what are there in the cytoplasm? What are there in the cytoplasm? Inside the cell, what are there? What are there inside the cell? In the cytoplasm, what are there? No cell wall membrane bounded organelles. I already told you. No membrane bounded organelles are present. Only the, what are those? What are those are present? Ribosomes, plasmids, nucleoid, only present. No mitochondria, no Golgi bodies, no endoplasmic reticulum. Okay, and 70th type of the ribosomes and glycogen bodies, which are reserve food material in the bacteria. Glycogen oil glands, especially PHP, polybeta hydroxy butyric acid. Very, very important. The bacteria storage food material, glycogen, that is PHB, polybeta hydroxy butyric acid. Polybeta hydroxy butyric acid. That is the reserve food material, especially that we are using in the preparation of biopolymer. Biopolymers preparation. Instead of plastic, we are preparing the bioplastic, degradable plastic by using these bacteria. This reserve food material also useful in the preparation of biopolymers. Okay, and you know the nuclear operators. That nuclear operators here, the DNA is only how it is, how the DNA, only coiled DNA is present, only coiled DNA is present. There is no membrane, there is no membrane bounded, there is no membrane bounded nucleus. Only coiled nuclei is present. That we calling it as nucleoid. Nucleoid. Also called it as genophore. Competitive exam, so many competitive exams they may asking genophore. Genophore. Next, only coiled DNA, which is present in the bacteria, also called as genophore. And nucleoid. Along with that, extra nuclear genome also present. Double helical, spirally coiled, self-replicated, double helical, spirally coiled, self-replicated, extra chromosomal, extra nuclear genome is called as plasmid. These plasmids are present in the bacteria. These are extra chromosomal genomes. What we call it as extra chromosomal genomes. Plasmids we call it as extra chromosomal genomes. That extra chromosomal genomes will be useful for the recombination and reorganization and genetical modification, genetical variation, and gene transfer. And in the gene transfer from one organism to other organism, we are using these plasmids are as a carriers, genetical carriers, gene carriers, the plasmids. We are using these plasmids, in the biotechnology, insulin production, so many anti. The Ledenberg plasmid name given to the extra chromosomal genome which is present in the cytoplasm of the bacteria, given the name as plasmid. By whom? By whom? Ledenberg. With the, the Jindal and Ledenberg, one gene, one enzyme, one gene, one hormone, one gene, one protein theory is there. That theory it is invented by Ledenberg to know the synthesis. Specifically factor plasmids. If the F factor is there, that we call it F plus. If it is not there, that we are calling it as F minus. The conjugation is taking place in between these two. The conjugation in the reproduction we will discuss. The bacteria reproduction, we will discuss it. The bacteria reproduction. Conjugation is taking place between the F plus and F minus. Which one having the plasmid, that is F plus. F factor, if it is having, that is F plus. If it is not there, that is F minus. There is a gene transformation from F plus to F minus. Is also having the plasmid, so that genetically modified gene transformation is taking place in between these two. And cofactors, coliphages, coli in the E. coli especially, coliphage which are present in the E. coli, and R factor, resistance factor, which shows the resistance, especially some plasmids, most of the plasmids, which some of the plasmids which shows the resistance to the antibiotics. Suppose plasmid F322 or PUC, these two F322 and PUC, two plasmids we are using in the genetical engineering. PUC, plasmid of University of California, first prepared this plasmid by University of California, that is PUC, and PBr322, that plasmid of Bolivia Rodriguez. Bolivia and Rodriguez, these two scientists first identified that plasmid, that is PBr322. These two plasmids, and PUC and PBr322. These two plasmids we are using in the genetic engineering for the gene transfers, which shows the resistance to ampicillin and tetracycline. Resistant zones are there in the plasmid. And tumor-inducing plasmids, the Agrobacterium tumefaciens. Agrobacterium tumefaciens that we are using, Agrobacterium tumefaciens we are using for the gene transformation in the plants. So many competitive exams asked which plasmid we are using the gene transformation in the plants. Are you there? Which plasmid we are using for the gene transformation in the plant? TI plasmid. TI plasmid, that is from Agrobacterium tumefaciens. Agrobacterium tumefaciens. That plasmid name it as TI plasmids. That is from Agrobacterium tumefaciens. These TI plasmids we are using for the genetical transformation in the plant, high yielding variety of the plant, disease resistant variety plant, alkaline resistant plant, salinity resistant plant, drought resistant plant, for like that, high flower shower tomato, like that. For the genetical modification transformation in the plant, we are using TI plasmids. Okay, and